Okay, so last time we were talking about major neurotransmitters, and I mentioned that a single neuron can release many kinds of neurotransmitters, okay? It doesn't have to be just one kind of chemical, and a single chemical can be used as both a neurotransmitter and a neuromodulator. Remember the difference? Neurotransmitters are chemicals released at the synapse, and neuromodulators are chemicals released basically anywhere else. But again, as I said, the same chemical can be both released at the synapse and into other parts. It can be released from other parts of the cell. And a single neuron can create and release many, many, many kinds of neurotransmitters. Okay, so now, once released, these chemicals, neurotransmitters and neuromodulators, they can act on other cells in different ways based on the receiving cell's receptors. And there are two broad categories of neurotransmitter receptors. So, two broad categories of neurotransmitter receptors. Okay, so what are these two categories? They are ionotropic, which we've already mentioned once, and metabotrophic. Okay, so let's define these. So our two are, the first one is ionotropic, which we already mentioned before. So you've seen this word. Where have we seen this word before? Do we remember? Okay, so maybe you'll remember once I tell you what an ionotropic receptor is. So these are proteins in the cell membrane, okay, that form an ion channel pore. Is this familiar now? Okay, and remember, an ion channel pore is a hole through which ions can cross the membrane. Okay, so when we were talking about the three kinds of electrical potentials, resting, graded, and action, we talked about ionotropic receptors, because these are often these are the ones that are most involved in those electrical potentials. Okay, so they are involved in both the electrical potentials and also in chemical signaling. So you'll see in just a moment. So proteins in the cell membrane that form ion channel pores, okay? And as a reminder, an ion channel pore, oops, I should have done that in purple. I'll just rewrite it. Okay, that form ion channel pores. And what are ion channel pores? They are holes through which ions can cross the cell membrane. Okay, so let's do a quick drawing. So we've got our cell membrane made of a phospholipid bilayer. Okay, so all right. Then we have our ionotropic receptor, which I will draw in black, I guess. Oh, I should have given myself a little more room. Okay. Okay, sorry about that. All right. Okay. And then we have our protein. And I'm going to give it this little empty spot, which you'll find out why in just a moment. Okay. And then we have the rest of the membrane. Okay. So this empty slot is waiting for some kind of neurotransmitter, so some sort of chemical messenger all right, so this is our neurotransmitter. So when a neurotransmitter arrives, and it clicks into place, this means that now the ion pore is open, okay? And this means that ions can now flow into or out of the cell, basically through the protein. It now has a channel. And remember, what were some of our most common ions being used in the nervous system? They are sodium, potassium, calcium, and chloride. Chloride is the only one that is negative out of these most common ions, okay? And so going back to ionotropic receptors in general, so usually graded potentials, they happen because of movement of ions through ionotropic receptors, okay? So usually these are the receptors that cause graded potentials, okay? And these kinds of receptors, they can open and shut really, really quickly. So they're involved in signals between cells that require a fast response, okay? And by fast, we mean milliseconds, all right? So, involved in signals between cells that require a fast, or let's say it requires a quick response, or fast, that's fine, that require a fast response because these receptors open and close on the order of milliseconds. So very, very fast, okay? So, ionotropic, very fast. They open and shut very quickly. The neurotransmitter acts as a key to unlock the tunnel, and through the tunnel flows ions, okay? So what about our second broad category of neurotransmitter receptor? So this second category is called metabotrophic receptors. Sometimes people pronounce it metabotrophic or metabotrophic. Okay, anyways. All right. So, in this case, in the case of a metabotrophic receptor, instead of physically opening a hole to let something in and out, they do something really different. Okay? So let's go ahead and draw our cell membrane. And we have our lipid bilayer. Okay, and now we have a protein with kind of a funny shape. There's a part of it that sticks out, and then it kind of wiggles a bunch of times across the membrane, and then it sticks into the membrane, okay? Or into the cell. All right, so what happens here? Our neurotransmitter in red is going to come and attach to the outside part of this protein. Remember, all receptors, when we say receptors in the nervous system, we're always talking about proteins, okay? So the metabotrophic protein, it doesn't open a hole. What it does is instead, when this gets attached, it creates a different chemical called a second messenger. And sends that into the cell. The reason why it's called a second messenger is because the neurotransmitter is considered, so this is considered the first messenger. Okay? And so then once the protein receives a neurotransmitter, it's going to create a second messenger. So this one is like, you know, you go up to someone's house and you knock, and instead of you walking into the house, you just pass them a note. Okay? And someone has to come receive it, and then they go do something. Whoever's inside the house is going to go do something else. Okay? So there are a couple of other things that it can do. All right? So these second messengers, they can create a new chemical that is going to go straight to the soma, and it's going to tell the cell body, create this other chemical. Okay? So this second messenger might go to the cell body and to the nucleus where chemicals are created, and it'll say, okay, I've received the message, we need to make this kind of chemical. Or this second messenger might be something that activates other ionotropic receptors in the cell membrane, and the second messenger will trigger the movement of ions in and out of the cell, okay? So metabotrophic, what does it do? Instead of opening a hole, these receptors, instead, they make either themselves or they make the cell create a signaling molecule, called the second messenger. Okay? Now the important thing to notice here is this is a much slower process, okay? So it takes much longer to activate this whole process, and a metabotrophic receptor may remain active for a really, really long time. And by this we mean on the order of seconds to minutes, okay? So these were milliseconds. You have 1,000 milliseconds in a second. So in the time that it takes for this ionotrophic receptor to open and shut 1,000 times, this one might be active only once, okay? So this is slow and more long-lasting, but also the signal can be stronger. Stronger signal, okay? And by slow, what do we mean by slow? We mean on the order of seconds or minutes. So it can last for a really, really long time. Okay, this section is quick. So we've now covered the main points regarding connections between cells inside of the nervous system. So next we're going to talk about connections to other body systems.